thank you to the organizers for this nice opportunity. I promise I will be fast. I'm going to skip some of what I had prepared, but I think that this is the important here. I'm going to talk about reprogramming, de-differentiation, and redifferentiation in vivo. Well, I don't think that I need this introduction here. Just mention that, as you know, Yamanaka and Takahashi, they reported the possibility of de-differentiating differentiated cells and bringing them back to iPS, the induced pluripotent stem cells, but this has been done in vitro. And the question we try to address is it possible to do this in vivo? One thing that is necessary in vitro, and I think that this was also very elegantly illustrated by Austin, is that you need a special medium to maintain the iPS. And so the assumption, and it was my idea, is that it was impossible to create iPS in vivo because the medium is not the right one. You don't have leaf in principle, and you don't have the right conditions. So the purpose, of course, is not to create iPS in vivo. That is not the final goal. The final goal is to try to take advantage of this possibility to do regenerative medicine in vivo without the requirement of ex vivo manipulation, ex vivo differentiation, and grafting. The grafting is a big problem that doesn't work at all. It's still a big bottleneck. So we created this version 1 of our reprogrammable mice. Other laboratories have created reprogrammable mice. In our case, we analyze what is the impact of this in vivo. This version 1 is ubiquitous expression of this cassette. This is a cassette that we obtained from Jenny's laboratory, and this is in the ROSA 26 RTTA. The cassette is randomly inserted in the genome with a lentiviral integration. We isolated 2 lines, A and B, and they are inserted. The cassette is inserted into these 2 genes, and we show that these genes are not affected by the integration or by the induction of the cassette. The mice are deposited in the Jackson laboratory, so are available to the community and to all of you, of course. Let me summarize here, for the sake of time, the phenotype of these mice when we treat them with doxycycline. So when we treat them with doxycycline, the adult mice, the cassette is expressed essentially everywhere. And the first phenotype that we observe is that there are dysplasias everywhere. This is diffuse dysplasias in many tissues. This is not surprising. One of the genes that is here is MYC. The other thing that is very interesting, and I will show you this data more in detail later, is de-differentiation. Cells lose their identity. They acquire a different identity in focal areas. This is also, the efficiency of this is low, as it is low the efficiency in vitro. In vivo, there are widespread expression of the cassette, but only focal areas of de-differentiation. And the other thing that we find are circulating iPS in the blood. After 1 to 3 weeks, depending on the dose of doxycycline, we have to stop the treatment because the mice stop absorbing nutrients. One of the most affected tissues is the intestine. So if we continue, there is malnutrition and the mice die of malnutrition. So we remove doxycycline, and then something very interesting to me happens, that is that everything goes back to normal. In about 2 weeks, everything redifferentiates. All these cells that had lost their identity go back to and redifferentiate, tissue recover homeostasis. I will show you this in a second. In the long term, of course, other things happen. Not everything redifferentiates correctly. A few cells, a few clusters of cells keep growing as iPS, and they form teratomas. Every mouse develops between 20 to 50 teratomas, representing a small percentage of cells that did not redifferentiate correctly. We also observe some tumors, different types of tumors, indicating that aberrant or fail reprogramming can induce tumors. We also observe IPS, and we also observe embryo-like structures. I will go in detail about this. So let me go here to the differentiation in the short term. So this is an example of what happens in the intestine and in the stomach. Here we are using double immunohistochemistry for a lineage, for an identity marker, cytokeratin 19. You can see here in the large intestine, this is the large intestine, you can see here one crypt with cytokeratin 19, but the next crypt here has lost cytokeratin 19 in part of the crypt. These cells have lost their identity. We can say that, I think, because they have lost cytokeratin 19, and some of them are expressing NANOH. NANOH, as Austin Smith mentioned, it was a gene discovered by his laboratory, is a protein that is a marker of cells that are very close to pluripotency, either pluripotent or very close to pluripotency. And you can see here that there is a variety of cells at different stages. Some express NANOH, others do not express NANOH. The same happens, we can observe the same in the stomach. These are glands in the stomach. You can see here one gland that has lost cytokeratin 19 altogether, and here there are cells with different levels of NANOH. Similar observations have been made in the kidney, in the pancreas, and in the liver. So the 4 Yamanaka factors have the ability to impose the transcriptional program in the physiological environment and to impose a pluripotency-like, I'm going to say pluripotency-like program, because we don't know if these cells, exactly these cells, are really pluripotent. As I said, when we take off doxycycline, everything goes back to normal. This is very dramatic in the intestine. You see here the dysplastic intestine. Some of these cells here are the ones that have lost their identity, but everything goes back to normal in 2 weeks. In the pancreas, it's even more dramatic. All this pancreas is dysplastic, is abnormal. The only part that is normal is here. In 2 weeks, everything goes back to normal. The laboratory of Yamanaka has reported this year essentially the same mice with very similar phenotype. They have characterized this redifferentiation more properly, and they have concluded that this redifferentiation happens without cell death. So the same cells that they differentiate, then they redifferentiate, either because they maintain some epigenetic memory and or because the environment is directing them to the proper differentiation lineage. We never observe liver in the intestine or intestine in the pancreas and so on. Everything goes back to normal. So this would be the advantage for regenerative medicine if we are able to control this, and this is where we're putting efforts. If we are able to control this, it would be possible to rejuvenate, in a way, tissues. So going back to the question, is it possible to induce the differentiation? Yes, it is possible. And of course, we need to find the right conditions to avoid tumors and teratomas. The main message I think of this work is that the in vivo conditions are permissive for reprogramming. Contrary to expectations, in vivo conditions allow reprogramming. And as I said, we can even isolate IPS from the blood. I will show that in a second. And the laboratory of Yamanaka has reported similar idea. And I want to take this message to take a few other messages from the literature to to propose a hypothesis. One is that people working in vitro reprogramming have found that innate immunity signaling, like TLR receptors, this is the receptor that detects double-stranded RNA, and probably others of the same family that are involved in detecting pathogens, they actually enhance in vitro reprogramming a lot. So every time we are doing reprogramming in vitro, we are using retroviruses or lentiviruses, and they per se are contributing to this reprogramming together with the Yamanaka factors. And the other thing is that IL-6. IL-6 is the paradigmatic inflammatory cytokine, is a member of the leaf family, and IL-6 also enhances in vitro reprogramming. The other piece of evidence I want to take is, was generated here at the CRG by Pia Cosma, and is the fact that damage in vivo induces reprogramming-like features. In the case of Pia, they induce damage in the retina, and some neurons that survive the damage express SOX2 and OCT4. And these are the ones that induce regeneration of the retina. And I want to make a package with this and speculate that perhaps reprogramming, the reprogramming that Yamanaka discovered, that has been considered until now a fantastic artifact not in the negative sense, but in the sense that it's totally artificial. It was considered something artificial that doesn't have a in vivo correlate. Perhaps we have to change our mind and consider that some kind of reprogramming happens when tissues are exposed to damage. And some cells are able to acquire a pluripotent or plastic state, and these are the cells that will repair the tissue. Among other things, because the in vivo environment is not that refractory to reprogramming. Actually, we think that is permissive, and we have shown that is permissive. As I said, when we induce the Yamanaka factors, we are able to isolate real IPS directly from the blood. This is blood that is extracted from the mice that are treated with doxycycline, but then we put the blood in the absence of doxycycline. So this is to capture reprogramming that happen in vivo. And these IPS have all the features of bona fide IPS. I'm not going to explain all of them, but they are real IPS. One of the things that we did was to do RNA-seq of in vivo generated IPS, of the classical in vitro generated IPS, and real ES cells. And when we did the RNA-seq, the computer we did hierarchical, unsupervised hierarchical clustering, and we found, this was one of the first surprises, that all the in vivo IPS, they were intermingled, mixed with the real ES cells. As you know, ES cells and in vitro IPS cells have a few differences. It's not clear how functionally relevant these differences are, but they exist. It's possible to separate them. Here, the in vivo IPS seem to be indistinguishable from the ES cells. Even though they are indistinguishable in the global RNA transcriptional profile, they are still different. And the first clue that they were not standard ES cells, they are not like ES cells, came from the following observation. When these mice develop teratomas, as I said, we induce the reprogramming factors for 2 weeks, and then months later, teratomas appear. These teratomas had something unusual, and was the presence of placenta-like differentiation. Of course, they had the 3 germ layers, they had ectodermal, mesodermal, and endodermal differentiation, but in addition, we saw placenta differentiation. As you know, placental differentiation is something that is forbidden in theory for standard ES cells under normal conditions. They cannot make placenta. And these are stainings with placenta markers, here cytokeratin 8 and PL1. And you can see that this is real placenta with the blood and everything, trophoblast giant cells and so on. Also, when we take the in vivo IPS that we have isolated in vitro, and we have expanded them, when we put them back into a mouse and generate a teratoma, they also generate this placenta differentiation. So it's a property that is maintained ex vivo. As you know, during development, the oocyte, the zygote, and the morula are totipotent. The first decision that cells make is to become trophectoderm or the inner cell mass. This is the first decision. The trophectoderm is going to produce the trophoblast giant stem cells and the placenta. And the inner cell mass is going to produce the embryonic stem cells, and so on. So in theory, embryonic stem cells cannot produce placenta. The assay that we did to test this more rigorously was to label the in vivo iPS with GFP and do aggregations. I have to emphasize that we are injecting here 10, 15 ES cells. We have not done this with a single cell. Some people have asked me that, have you done it with a single cell? Well, it's very difficult, very, very challenging, very inefficient. So we are putting here several cells. So perhaps there is a heterogeneity in the culture. Some cells have some properties and others other. We have not figured out this yet. But they come from a clone. They are clonal cells. When we do this, then we test where these cells go in the blastocyst. Here are ES cells. ES cells go to the inner cell mass, no surprise. But these in vivo iPS cells, you can see that in addition to the inner cell mass, they also go to the trophectoderm. You can see one cell here, you can see another cell here. Still, these cells may not be functional, but they are. If we put these blastocysts into a mouse, into a foster mother, sorry, we obtain embryos where the embryo is GFP positive and the placenta is also GFP positive. So they are contributing to the placenta. And this is something that doesn't happen with normal ES cells. So these cells that have been generated in vivo, for some reason, they can do this transition to the placenta. Either we call that totipotency-like features. I don't want to say that they are truly totipotent, because real totipotency would be to generate a mouse directly from these cells, something that we have attempted, but we have not achieved yet. Still, to go one step ahead, these assays have been done using a morula as a scaffold. Remember that we are putting these ES cells into the scaffold of a morula to generate the embryo, the blastocyst and the embryo. So we wanted to avoid that altogether and try to get something similar to an embryo directly from these cells without the help of real totipotent cells. And for that, what we did was to inject the in vivo iPS and in vitro and ES cells, to inject them intraperitoneally. We have tried to do it in the uterus of pseudo-pregnant mothers to recapitulate a real pregnancy, but all that has failed. The reason why we did it in the peritoneum is because it's known to be a place permissive for pregnancy, aberrant and pathological pregnancies, but it's possible for an embryo to develop to some extent in the intraperitoneal cavity. So we failed in the uterus, so we went to the peritoneum. And when we injected these cells in the peritoneum of these mice, of wild-type mice, these are not reprogrammable mice, these are regular mice, we observed with low efficiency these kind of vesicles that were floating in the peritoneum. They were not attached to any tissue. And we were very surprised by the histology of these structures. They are definitely not teratomas. Teratomas, as you know, are compact masses with differentiation of all the three germ layers. In this case, it's a complex structure with layers. And actually, when we did immunohistochemistry, these layers are organized in a manner that could resemble embryonic structures. Here is SOX2, ectoderm, brachyury, mesoderm, and GATA4 endoderm. But more surprising than that, at least to me, was the presence of extraembryonic structures. Remember that ES cells should not produce extraembryonic structures. We have here CDX2, a marker of trophectoderm, and we have here markers of extraembryonic mesoderm, the yolk sac. Actually, the yolk sac is the one that produces the first erythropoiesis in these so-called blood islands. You can see one here, but I will show you more in detail. This is another structure, another of these structures, and you can see here endothelial cells in this yolk sac-like structure that these cells, by themselves, without the help of any other cell, can produce in the peritoneum. When we do this with in vitro IPS, we have never obtained these structures, and when we have done this with real ES cells, different clones, we have never observed these structures. So these structures, we have only observed them at low efficiency with this in vivo IPS. Of course, injecting 100,000 cells or 1 million cells. So it's something that happens with low efficiency. But it seems to be a proprietary, exclusive of this in vivo IPS. This is another structure. And actually, this is consistent with the ability of these cells to contribute to the trophectoderm and to the placenta when we do aggregations in morulus. That is something that happens with very high efficiency. And with this, I'm going to finish thanking the in vivo reprogramming team, especially Maria Abad, who was the person who spearheaded this project. And I will be happy to take questions. Thank you.
Yeah, you made this remark about using this